Namaste, myself Jimmy, Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we will discuss about the requirement of plant biotechnology lab. It will include also plant tissue culture lab requirements. So before starting about the plant tissue culture or plant biotechnology labs, we have to know that what are the accessories, what are the instruments, and what is the basic infrastructure should be completed or should be there in the lab, so that we will not get any kind of a problem or issue in the plant tissue culture lab performance. So need of low cost lab. Low cost option should lower the cost of production without compromising the quality of the micropropagation in plants. Micropropagation technique is too much costly. Potential increasing interest in agriculture crop production increased. Low cost tissue culture technology will stay a high priority in agriculture, horticulture, forestry and for floriculture of many developing countries of the production of suitably priced high quality planting material. General requirement for the setup of a low cost laboratory. Here is the flow diagram where you can simply see this is the entry region where you will get the bench, and it will have sink and then bench for the media preparation area. This is the chemical store. This is the analytical instrument where you will perform autoclave will be there over there, pH meter, your microscope and microwave balancing over there. This is the photography or dark room, and this area is sterilization area. This is airlock area. This is hatch. This is the dissection room where you will perform the inoculation process, and this is the wash up room, and this is the wash up room where you will get all the clean glasswares. And finally, after the inoculation of eggs plant into the tissue culture media or flask, you will put all the cultured media into the culture room with shelves, and this is the airlock and this is the exit area. So this is the flow diagram of plant tissue culture laboratory. Now come to the location. The location shall conform the land to the land use plan of the T of the area. The building should be located away from the sources of the contamination such as soil mixing area, storage, etc. The site shall be accessible to service road, water supply and electric lines. The site shall be well drained. And the structural requirements, in the structural requirements, the roof structure should be treated timber or steel with anti-rust paint. Roof vents when provided shall be properly screened. Ceiling shall be at least 2.4 meter from the finished floor line and painted with latex paint and should be prevented from the dust. The wall shall be concentrated and smooth finished and properly painted with antifungal epoxy paints, and slope shall be at 45-degree temperature. All windows shall be properly installed with 16 mesh screen and at least 1 meter from the finished floor line. Main doorways shall be at least 1.5 meter wide and moisture and rust resistant material. Floor shall be concreted, smooth, non-skip slope 2% to 4% towards the drain. The basic facilities of the laboratory: one room with computer with locker with for keeping apron, working place, layout of the plant tissue culture, washing room, sterilization room, media preparation room, inoculation room, incubation room, skilled labor. In the washing room, the glassware washing area shall be located near the sterilization and media preparation area. Washing sink can be fixed or fitted in one side of the room, should be acidic and alkali resistant. Room are also provided with bucket, detergent, mug and also plastic tray to keep washed media, bottles and sterile media. Room provided with water line and electricity line. In the sterilization room, the room should be have one autoclave or cooker with well ventilated exhaust fan, also having tools for support while placing the media bottles in autoclave or cooker for sterilization. The bottle should be sterilized at 121 degree temperature at 15 pascals for 1 hour. In it contains wrapping papers to wrap and rubber to tight the inoculating dishes for sterilization process. And in the media preparation room, the area shall be provided with working tables which should be up to 1.2 meter wide and height should be 850 millimeter to 900 millimeter. Tabletop shall be covered with material which are easily cleaned and which will stand disinfectant solution. Water resource and glassware storage area shall be provided. Room contains double distilled water unit, pH meter, buffer solution, electronic balance, cooling freeze, measuring cylinder, magnetic stirrer, pipette, hot plate, media bottle. Room should contain the stock A, B, C, D, E, and F solution with required chemicals such as sucrose, adenine sulfate, inositol, agar-agar, which is solidifying agent, etc. In the inoculation room, the floor of room covered with tiles of facilitated proper cleaning, and room contain the laminar airflow cabinet with installation of HEPA filters for filtered air supply. It also requires sodium hypochlorite, mercury chloride, and 70% alcohol for surface sterilization. It requires forceps, scalpel, sterile bottles, sterile dishes, spirit lamp, cottons, and hand care, etc. In the incubation room, width of racks should be 405 millimeter to 1 meter. The distance between each layer of the rack should not less than 455 millimeter. Photo period racks with culture bottle shall be provided. We mostly operate light intensity with a standard day length of 16 hours and 8 hours dark regime. Temperature is maintained at standard temperature 25 plus 2 degree temperature with 1.5 tone window AC to creating the control environment condition. Light intensity required about 200 lux to 1600 lux for growth of culture. In the instrument room, bottle washing machine and autoclave freezer and rotatory shaker, multiple purpose magic stove, weighing balance, thermometer, magnetic stirrer, digital pH meter, laminar airflow with UV tube, rack photo periodic culture, glass bed, sterilizer, air conditioner with stabilizer and micro air oven, etc. Glasswares and other requirements, so glass bottle, petri dish, burette, funnel, back stand, pipettes, conical flask with screw cap, graduated measuring cylinder, spatula, scalpel and forceps, wrapping papers and bucket gloves, etc. We need, and practical manual of plant tissue culture should be available over there. And in the other facilities, we need hand washing facilities, fire exhaust extinguishing system, waste disposable area should be provided over there. So this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.